the acquired immune system. Okay, here's your objectives. All right, so as I mentioned before earlier this morning, this is your very sort of specific, sophisticated immune system, right? We know that the second line of defense is inflammation. We know that it's very effective. It's fantastic. But it's not specific, and sometimes it's not enough, right? We need that third line of defense to kick in. And that is where now adaptive immunity comes into play. So the purposes of adaptive immunity is, one, destruction of infectious microorganisms that are resistant to that second line of defense. And two, it's good and helpful because it has long-term, highly effective protection against future exposure to the same pathogen. So it has memory. That's why it's very specific and more sophisticated than inflammation, okay? We also know that it's inducible. And what I mean by inducible is that it sort of recognizes pathogens as non-self. And it's also specific, meaning that among many pathogens, a unique pathogen is identified and destroyed. Okay? So that's what these sort of terms up here on the screen mean for you. Now, the end products of adaptive immunity are bifold, so to speak. You have immune globulins or antibodies, and you have lymphocytes. So those are your T cells and your B cells. So these are the end products of the adaptive immune system. And we're going to talk more specifically about those from just a second. How about immune globulins first, though? Antibodies. Where do they come from? Who makes antibodies? What cell of our immune system makes antibodies, makes immune globulins? Anyone know? B cells. Our B cells make immune globulin, which, again, can be interchanged as antibodies. Good. Every specific B cell and every specific T cell in the body fights one specific type of pathogen. Now, that might not sound very effective or very efficient, right? That only one particular type of B cell can only fight one particular type of pathogen. However, when you put all the different types of B cells and T cells together, they actually can cover billions of microorganisms. So it actually can be pretty efficient and pretty effective. And this term of sort of these specific different functions of all these B cells coming together to create this universal protection is a term called clonal diversity. And we're going to talk about that more specifically in a little bit, but I'll write it up here on the board. Clonal diversity is sort of what refers to this ability of all these different types of B cells and T cells coming together to create universal coverage. Okay? So the end products. Let's talk more specifically about those. The first one I mentioned are immune globulins or antibodies. These are produced by B cells, as I mentioned. They do constantly and forever sort of circulate in our blood. And the way that they work is that they actually bind to the antigen on the infectious agent. So remember, antigens are bad, right? Antigens are the bad guys. And these immune globulins or antibodies bind to those antigens from infectious agents. Antibodies can actually result in direct inactivation of the microorganisms by specifically directing and sort of killing those pathogens. Or they can actually activate inflammatory mediators that will end up actually destroying the pathogen. So what I want you to sort of get from that is antibodies can either directly kill the pathogen, or they create an immune sort of mediation response that then will eventually destroy the pathogens. To be clear, when we're talking about antibodies, we refer to that sort of branch of this adaptive immunity as humoral immunity, okay? So it has to do more with the antibodies themselves than the actual B cells. Now, let me make that clear for you. Do B cells actually kill and attack pathogens? No. B cells do not directly have an impact on the antigen and or the pathogen. It's the antibodies that they make. It's the antibodies that they secrete that actually kill the bad guys. B cells, again, do not directly attack antigens or the pathogen. Yeah. so they sort of go hand in hand. That's a great point to bring up. I don't want you to think of these things as happening sort of independently in silos from each other. There has to be very close communication between the second line of defense and the third line of defense. And we'll talk more about that relationship and that communication between them later. Yeah. So B cell production of antibodies and the actual effect the antibodies have on the antigens are referred to as humoral immunity, as opposed to the T cells. And there's a lot of different subpopulations of T cells that we're not going to necessarily get into today, like your effector T cells, your killer T cells, all of those T cells. We're not going to necessarily talk specifically quite yet about. But that's what we're talking about when we're talking about the T cells. They are different than the B cells in that they don't secrete something like antibodies to kill the pathogens. They kill the target directly. So that's a big difference between B cells and T cells. They can also stimulate other leukocytes through cell-to-cell contact or secretion of those chemical messengers, those cytokines, to work against the target as well. And T cells are referred to as the cellular immunity of the adaptive immunity. So the cellular immunity arm. Now, to be clear, both produce memory cells. So both, no matter if it's the antibodies or from the B cells or the T cells, they both can have memory, which is super helpful because now with memory, the second time around, the response is much quicker. So now you've had one particular pathogenic invasion. The B cells and or the T cells have come to the area, have killed the target. Now, forever after, they release these sort of like surveyors, so to speak, or these surveillance systems that float around in the blood. So the next time that you're infected by this same pathogen, the response is that much more effective and that much more quick because they had memory to that specific antigen. And they interact. So B cells and T cells, the humoral arm and the cellular arm of immunity, can work together and interact together, to be clear. And it's this collaboration between B and specifically helper T cells that's essential for almost all of those antibody responses to antigens. So they do work very well together. Okay. And again, remember that each individual B or T cell has a specific, specifically recognizes one type of antigen, and that is called clonal diversity. Okay? So let's talk some terminology now. Let's talk about the different types of immunity that can sort of be elicited under this sort of umbrella of adaptive immunity. So the first one is active immunity. So active immunity is when the person, the patient, the individual, whatever you want to sort of think about, has a natural exposure to an antigen. Okay? So this could be a natural exposure to a vaccine, an immunization, or it could be the fact that you actually had the disease. You actually got the infection. Either way, you've actually engaged the immune system, and they have fought, the immune system has fought the infection. Active immunity is permanent, right? You've engaged the immune system, the B cells, the T cells have responded, and now either because you got a vaccine or because you've actually had the infection, and now you have permanent immunity after that. Again, because of those memory cells. That's active immunity. If you've had the disease or you've had the vaccination, that's an example of active immunity. As opposed to this other term, passive immunity. So passive immunity happens when preformed antibodies or T cells are just simply transferred from donor to recipient. Okay? And this is a very stark contrast to active immunity in that with passive immunity, the immune system of that individual is actually not engaged at all. The immune system is not active at all with passive immunity. It simply just accepts some other cells from a donor. Okay? Anybody know of any examples of passive immunity? Yeah. Colostrum. Very good. So breastfeeding. Very good. The antibodies pass from mom to baby. Very good. Can you think of any others? Good. Blood transfusion can be for a short term. Yes, that's very true. Especially like when you're talking about like blood transfusions or infusions for immune type purposes, not just for sort of like, someone gets a blood transfusion after surgery, but right, yeah. Other ones, yeah. Could be, yep. Bone marrow transplant. Pregnancy. So there actually can be antibodies that go from mom to baby through the placenta. And the last one, yeah, that I was going to say. Go ahead. No, go ahead. Yes. So, right. So the other example I was going to give are some types of immune globulin that they give for contact from certain things. Right. So an example could be like rabies. That's an example. Rabies immune globulin, like if someone got bit by a rabid dog or something, they could actually give antibodies, preformed antibodies to the recipient to sort of protect them from rabies. Another really good example is hepatitis A. There's a pretty good hepatitis A immune globulin as well that can be given if someone came into contact with hepatitis A. Again, is it forever? Someone who gets immune globulin, hepatitis A immune globulin, are they forever protected from Hep A? No, it's temporary because it's the donor's antibodies or T cells are eventually going to get destroyed by the host, by the person who's receiving it. So it's a temporary protection. That's the difference between passive and active immunity. Is that clear? Does that make sense? Okay. All right, some other terms. Antigens, as I mentioned, are the bad guys. They bind with the antibodies or the antigen receptors on the T and B cells. And then immunogens. So immunogens are the antigens that actually induce an immune response. So that being said, immunogens, all of them are antigens. But antigens aren't necessarily immunogens. Why? I'll say that again. Immunogens induce an immune response, and they're all considered to be antigens. But antigens aren't necessarily immunogens. Why?
They can detect self and non-self. So
okay.
Cells can detect other cells and
Okay, good. That's very true. And the other thing I would sort of couple with that and add to that is immunogens, by definition, induce an immune response. Now, there could be some antigens that are foreign. They're the bad guys, but they don't adequately induce an immune response, either because there's too many of them that sort of overload the immune system, or there's not enough of them that doesn't mount a proper sort of full-blown immune response. So they have to actually induce an immune response to be immunogens. So they're all antigens. Both antigens and immunogens are antigens. But not all antigens adequately induce an immune response, so therefore those specific ones can't be called immunogens. I hope that makes sense to you. The success of an acquired immune response depends on the function of both the humoral and the cellular responses, so the antibodies and the B and T cell responses, as well as appropriate interaction sort of between the two of them. If that doesn't happen, then you don't elicit an immune response and you don't get immunity to that particular antigen. Okay. And sometimes it's actually not only the quantity of the antigen that either adequately or not adequately induces an immune response, but can also be things like the size or the sort of the quality, so to speak, of that particular antigen, of whether or not it actually elicits an immune response from the immune system. Okay, so to be clear, just because you have an antigen in the system doesn't necessarily mean that you become immune. Doesn't actually mean that you have a proper immune response to it. And that's what I want you to understand from this. Okay. So that being said, certain criteria certainly influence the degree to which an antigen is immunogenic, and I sort of mentioned that. So how foreign is it? So foreignness to the host. That's sort of one of the biggest things, is how abnormal, how foreign to the body is it? Adequate size, sufficient quantity, etc. There are also some other factors in terms of the actual individual that can dictate how immunogenic an antigen is. So how did it get in? So its entry route. Genetics can certainly play a part, so genetics of that particular individual. There is a very sort of small percent of the population that may fail to produce a sort of measurable immune response to some vaccines. Despite getting vaccinated several times, they just don't ever seem to become immune to it. And there, again, it's sort of a small percentage of the population, but that's a good example of genetic factors dictating how, what kind of immune response is mounted. And then, is there other diseases going on, other drugs present, et cetera. Okay? How do we know whether or not someone has become, the degree in which someone has become immune, how well their immune response is to an antigen? How could we find out clinically? Right, titers. Right, testing, doing a blood draw for titers of whether or not how well their memory cells are present, or how much memory cells are present and how immune they actually are. So how many antibodies are in the blood, so to speak, for a particular type of antigen can tell us and can clinically test how well an antigen has elicited an immune response. Good, very good. Okay. So let's talk now for the next few minutes a little bit about both the humoral immune response, so how the antibodies specifically respond to create immunity, and then how the cellular immune response actually elicits an immune response. So let's talk first about immune globulins, okay? Antibodies is the other term for that. As I mentioned, these are produced by the plasma cells that mature from lymphocytes, and specifically they're produced by B cells, as I mentioned. There are different types of antibodies that sort of differentiate by the difference in sort of the structure and the function of what that particular antibody does. There's a couple that I really want you to be familiar with. The first one that I have listed here is immune globulin G. Anyone remember this or talked about immune? Okay, good. So this is kind of a review. Immune globulin G is for sure the most abundant, especially in secondary immune responses. So especially the second time someone's exposed to that particular antigen, these really are there. They're really abundant, especially in that second immune response. Immune globulin G accounts for most of the protective activity against infections, and it can actually have selective transport across the placenta between the mom and the baby. Immune globulin A, we're not going to spend a lot of time with today, but it's often found a lot in our secretions. So in our saliva, in our tears, in our GI secretions to help sort of prevent infections in some of those tunnels and some of those tracks in our body that we talked about. I'm not going to spend a lot of time about that today. Immune globulin M, though, is the next one that I really want to spend some time talking about. It is the first antibody that is produced during an initial response to an antigen. So the two big ones that I want you to remember are immune globulin G and immune globulin M. And how I want you to remember them is that immune globulin M is the first antibody produced, and immune globulin G is the most abundant, especially the second time someone's exposed to that particular antigen. Okay? Now, when talking about sort of the function and the way that antibodies work, we have to talk a little bit about this antigen-antibody binding. Now, I'm not going to spend a lot of time on this slide. You can sort of read it. But what I truly want you to just sort of take away from this slide is that antigens fit into the antibody binding site with the specificity of basically a key into a lock. So again, there's a specific antibody for every antigen, and they fit together perfectly like a key into a lock. That is how antigen and antibodies bind for the antibody then to sort of digest and destruct that particular target. And that's really all I want you to understand about this slide. Okay. Antibodies can protect, obviously, against infections either directly or indirectly. Now, the way that they can do it directly is something called neutralization, where it literally just blocks the binding of the antigen to that particular cell receptor. And I have a picture of it in this next slide. So here you see, this is virus, just as an example of virus neutralization. So here's the virus, and here are the antibodies, right? So in this particular case, they just bind directly, this little Y piece here binds directly to the virus, so that when it tries to come into contact with this cell receptor on this particular body cell, it can't. It's blocked. It doesn't fit into the lock and key anymore. That's exactly how neutralization works, okay? And that's what I truly want you to understand about the direct protection against infection. Now, there are other ways that antibodies can indirectly affect the sort of immune response, and that's through things like the sort of the relationship with that second line of defense. So antibodies do work in communication with that second line of defense in the inflammation process, in phagocytosis, and in that complement plasma protein system that we talked about. And again, we talked about how well that antibodies protect us from whatever particular antigen this is. There's varying degrees, we know, from person to person, and the way that we truly know how effective it is by getting that antibody titer, by actually drawing blood, okay? And that's truly how the antibodies function to protect us. Many pathogens initiate infection by attaching to receptors on cells, and antibodies block that attachment of antigens onto those cell sites. Okay? All right, and there's a good picture for you there. Okay. And then cellular immune response, those are your T cells, right? So as I mentioned, there's a lot of different types of T cells. We have the T cytotoxic cells that attack antigens directly. And then you have the T helper cells that actually help control both cell-mediated, so the work of the T cells in that immune response, but also works with the antibodies that are formed from the B cells in that humoral arm of the immune response as well. And then one of the most sort of specific, sophisticated components of T cells is that what we've already talked about, the ability for them to remember, that they produce memory cells. They remember an antigen that's been previously seen by the immune system, and they induce a secondary sort of immune response that is so much quicker and so much more effective the second time around. So I kind of think about it, as I mentioned earlier, as they sort of secrete these like security guards, right? And they are just constantly floating around in the bloodstream. So the next time that it comes into contact with this particular infection, with this specific pathogen, it elicits that much of a more quick, efficient, effective response. So they're very smart, very sophisticated in that way. Okay. Do you understand the difference between humoral and cellular mediated immune systems, immune response? Yes? Okay. Okay. And then for the next little bit here this morning, we'll talk a little bit about how the humoral, again, to be clear, what is humoral? When we're talking about the humoral immune response, what are we talking about? Antibodies. And the cellular immune response is who? T cells, right. So let's talk now a little bit for the last couple of minutes here regarding the collaboration of these two branches together, because they do work together, and they do respond sort of in two different phases together. So the first one is, again, that mention of clonal diversity, right? That the way that, I mentioned that the specific B and T cells have specific sort of antigens that they work against. And that production of that sort of specificity, that sort of what these B and T cells are created to do, happens before birth. It actually happens and is sort of selected out prior to birth, so in utero, in the embryo. This whole sort of ability of specific antigen recognition. Then what happens, again, this is before birth, is that the maturation of the T cells and the B cells occurs in specialized lymphoid organs in our body. So specifically, the T cells come to mature and come to maturate in the thymus, so it's kind of easy to remember, T cells in the thymus, starts with a T. And B cells come to mature in the bone marrow. So again, B cells, bone marrow, B. Kind of easy to remember that way. And what happens when these different types of cells mature in these different lymphoid organs is that it results basically in this ability in differentiation of these lymphoid stem cells into B and T cells that create this specific reaction against any antigen that can be encountered throughout life. Again, that is clonal diversity. That occurs, again, to be clear, I will say it again, this occurs before birth. Okay? Then what happens after birth and throughout life is something called clonal selection. This is more antigen initiated. So this is, what happens after birth and throughout life when these sort of branches of the immune system come into contact with antigens. And now what happens is that you have this sort of complex cellular interaction in lymphoid organs that then elicits this response for these either B cells or these T cells to actually respond to their specific type of antigen. And that is termed clonal selection. In clonal selection, B and T cells have to really sort of be officially presented with that specific antigen before being able to sort of destroy it if it's a T cell, or being able to sort of create antibodies against it if it's a B cell. And there are specific types of cells that we're not going to necessarily talk about today, but there are specific types of immune cells that basically, for lack of a better way of explaining it, present to the B cell or present to the T cell on this silver platter. Like, here is your antigen, your specific antigen, now go, counteract it. And that's what occurs specifically in clonal selection. All right. As I just mentioned, this clonal selection is initiated when the T and B cells interact specifically with their specific antigen. And these antigens must first be sort of noticed, processed, and then presented to either the B cell or to the T cell by these cells called antigen-presenting cells. So that's the cells that sort of present to the B and T cells. Here's your antigen. Have fun. Go ahead and attack it. They're called antigen-presenting cells. Okay? Then once that occurs, B cells will then create their antibodies to attack it, or T cells will actually, directly attack that antigen. Okay? Now these next few slides are kind of a reiteration and kind of a summary of what I just said. So, for processing and presentation of those particular antigens to those B cells or to those T cells to occur, the antigen must be that particular antigen for that particular cell. So it must be the appropriate type, the appropriate size, and it must have that specific affinity for that sort of lock and key sort of situation for that particular type of cell. Okay? And these next few slides, so B cell clonal selection and then T cell clonal selection, are just kind of more technical, more little FYI slides for you to understand exactly how B cells get activated and how T cells get activated in this type of immune system. Okay, so you can just kind of read over those. You're not going to have to know much more specificity about those. Again, you see here the difference between the first time someone is exposed to an antigen. So you have your sort of initial exposure, and then you'll see this kind of latent period or this lag phase from the initial exposure. This is when the B cells are differentiating. And then after 5 to 7 days, you'll see that immune globulin N antibody for a specific antigen is detected. So again, immune globulin M and then immune globulin G will have either sort of an equal response that immune globulin M had, but come after immune globulin N. And then the next time around, that next time that someone is exposed to that happens a lot quicker. That lag time and that latent period is a lot shorter because now immune globulin G has that memory to it and will produce a larger amount of antibody to that site. So again, that's the sort of the difference between primary and secondary response, is that the second time around, the immune response is so much faster, it's so much more efficient. And the rapidity of that, the reason that it's so much faster is because of the presence of those security guards, those memory cells that are floating around in the blood. And then this is the last thing here. Immune globulin M is produced in similar quantities to the primary response, but it truly is that immune globulin G that is produced in way considerably greater numbers the second time around. And that, again, that affects into its really effective, quick response in that second time around. Okay? And then then last but not least, I always liked, if possible, to kind of give you a visual in terms of what you might see on the age continuum with whatever topic we're talking about. So now with this topic of immunity, I wanted to just list a couple of things here for you of some differences or some changes that you might see when talking about immunity in the pediatric population. So this kind of gives you some of that sort of FYI information there for you. And then the next slide here is for, is for the aged, so the elderly considerations with immune function. So that's just kind of some good sort of information there for you. And then last but not least, it kind of just discusses this interaction between lymphocytes and phagocytes for you. And then I really feel like, as I mentioned throughout the day today a lot of this stuff is very abstract. And by no means do I need you to understand all the specificities of it and the scientific, molecular sizes of antigens and all of that sort of thing. So really what I wanted to do here at the end for you is, in summary, kind of give you an overview of those 3 lines of defense that we talked about and really highlight the things that I want you to be familiar with and that I want you to sort of understand. So I have all 3 lines of defense here. So barrier, skin, inflammation, and then your more specific adaptive immunity. And each one of these here kind of lists for you the different sort of categories of their characteristics. So what kind of level of defense they provide, how long it takes, or kind of the timing of the defense. Are they specific? Are they not? What cells are involved? Do they have memory? Do they not have memory? And then what kind of protection is involved with that particular line of defense? This kind of breaks down some of the abstract concepts that we talked about today into nice little categories for you. And hopefully make some sense conceptually for you about comparing one to the other. So that is there for you for review as well. We've also talked about a lot of different terms today, so from mast cell to monocyte to phagocyte to all these different types of terms. You will also notice in Blackboard that I have a sort of a review of terms for you as well, just a document, just a sort of straight informational document that kind of is a review and reminder of some of the terms that were thrown out today, just to kind of give you some definitions and give you some context for those as well. So that's in Blackboard for you as well under today's dates folder. All right? And that is it, guys. Thank you so much. That's the sort of the broad sort of introduction to a more specific immune lecture we'll have next Monday. Have a great week, guys.